Okay, good morning, everyone, and welcome to another Earth Institute Coffee Morning. So we've got a double act today. So from two of our great associate members, Tony Twamley and Dirilgen, who have both won prestigious IRC Postdoctoral Fellowships, and they're going to talk about their work in agricultural and environmental systems. So Tony's up first, and he's going to be talking about the role of receptor-like kinases in defense priming in wheat. Tony.
Great. Morning, everybody. I'd like to thank Emily, Taz, and Will for inviting me to talk. I'd like to thank the Irish Research Council for funding my project and Dr. Angela Feakin and UCD for facilitating it. So there are many challenges to the production of wheat, not least fungal pathogens and the diseases that they cause. These can cause up to 20% losses annually. The current controls for these are chemical fungicides and resistant cultivars, but pathogens such as powdery mildew in the picture here can build up resistance to these controls. So we need to find other ways of protecting our crops. My project is investigating the role of receptor-like kinases, RLKs, in defense priming in wheat. So receptor-like kinases are like a sensor that can detect the presence of a pathogen. And defense priming elicitors are a type of treatment that can induce the plant into a state that makes it more ready for a pathogen attack. So plants and pathogens have co-evolved, and as a result, there's been this tit-for-tat constant arms race between them. Pathogens are always trying to gain access, and plants have developed preformed and inducible defenses against them. One such mechanism that plants use is receptors on their cell membranes, and these include receptor-like kinases. And these receptors can detect molecular patterns that come from the pathogen. Once they're detected, they can trigger an immune response. It's much like the sensors on your house, at your window and at your door that are connected to your house alarm. So like these sensors, they detect a burglar through vibration or motion and set off the house alarm. And in a similar way, that's what this type of immune response is. But some adapted pathogens have figured out a way to circumvent this immune response. But wouldn't it be great if we could increase the chances of the plant being able to detect the pathogen? And that's where priming comes in. So there's a difference between priming elicitors and fungicides. Fungicides have they basically are designed to kill fungal tissue, either systemically or by direct contact with the fungus tissue. Whereas priming elicitors are not antifungal. They stimulate the plant's own defense mechanisms, giving a more robust defense against the pathogen. And that reduces the amount of disease compared to a non-primed plant. At a molecular level, you would expect to see some gene expression of defense-related genes after the priming stimulus has been applied. Then you would expect to see that gene expression level to drop to normal levels. And then upon a challenge from a pathogen, you would expect to see a swifter, stronger response of gene expression of these genes to put up a strong immune response to that pathogen. So my project encompasses 4 aims. Firstly is to identify these RLKs. So I kind of had a leg up from my PhD work in that I've identified 21 that respond to the priming stimulus and then do what I expect, return to normal levels, and then respond again to a subsequent pathogen challenge. I want to shortlist these based upon if they're still activated by different biologically derived elicitors. That would be, say, chitin from fungi, harpin from bacteria, laminarin from seaweed, etc. Then I want to confirm the role of these RLKs in the priming effect. And I'll do this by silencing these genes and seeing, does the priming effect still occur or not? Then lastly, I want to optimize these elicitors by testing different treatments and also testing them against different fungal pathogens and seeing, can I optimize them for each fungal pathogen, etc. So that's my project in a nutshell. Thank you for listening.
That's great. Thanks very much indeed, Tony. That was really interesting and really clear. And I particularly appreciated the house alarm sensor analogy. That made it all make sense to me straight away. Thanks.
Okay.
So Tara, would you like to share your screen?
Yes.
Work away.
Okay, cool. Good morning, everyone. I'm delighted to get to give you a glimpse into my fellowship research that I embarked on at the beginning of the year. Hold on. Sorry, I just want to get rid of you guys here. You're in my way. So soil is alive and teeming with life. Biodiversity in soil is truly a hidden world, not least due to the opaque nature of soil, but also due to the size of the organisms within it. This infographic is from an FAO report that came out only a few months ago. And it shows on the left that the soil community is made up of organisms that range in size, from earthworms to bacteria and fungi. And the right illustrates that these interact with one another and the soil environment, contributing to the dynamic nature of soil. And for those of you who are like me and need a visual glimpse into this world, here are some of the small invertebrates found in soil, about the size of a full stop. And yes, I am intentionally showing you the cute and colorful ones. So global biodiversity loss may very well impair ecosystem functioning, if not already. And as soil biodiversity is a major component of terrestrial biodiversity, its loss has the potential to reduce most ecosystem services provided by soils, of which there are many. Above ground, insect pollinator diversity is important for providing pollination services, which is required by 75% of crops and over 80% of flowering plants globally. And actions to increase farmland biodiversity are promoted in Ireland nationally through agri-environmental schemes and other conservation initiatives, such as the All Ireland Pollinator Plan. And one of these measures include allowing or facilitating wildflowers to grow on farms by adopting practices such as the field margin that you see here. Non-crop plants are quite important because insect pollinators such as bees also need to forage on wild plants outside of the crop's flowering in order to complete their life cycle. So while the value of biodiversity to agriculture is being increasingly recognized, there's also the finding that what happens below ground can have impacts on how plants respond above ground and vice versa. And there's a decade or more of research looking at this in the scientific literature. Not yet fully explored though, is how below ground soil communities might be indirectly affecting above ground pollinator communities by altering floral rewards and traits, and how these might be affected by agricultural management practices. So cue my IRC research. As already mentioned, there's a limited work out there integrating below ground ecology with pollinator ecology, and those that have mostly looked at single species inoculations or focused on a single taxa. So this project will explore soil plant pollinator interactions with a multi-species view to account for the fact that there are many below ground interactions taking place in nature. It will look at the effect of soil biodiversity on the plant, both the visible part of the plant that the pollinator interacts with, but also the hidden part, the roots. And roots are a whole world in themselves, really, and they have a dynamic relationship with the physical soil environment and the soil organisms. So they're much more than just anchoring the plant. It will also investigate the effects on the behavior of pollinators. This is of interest because pollinators rely on visual and olfactory cues when visiting plants. So any bottom-up effects could be ecologically and economically noteworthy. Lastly, these interactions will be further explored in the context of agricultural management practices. Pesticide use is one of the key potential drivers of pollinator decline. And many studies have looked at the sublethal effects of exposure on wild pollinators. More recently, though, pesticide residues have been found in wild plants growing in field margins and in soils. Again, it'll be interesting to explore the effects of pesticides on organisms in soil and its knock-on effects, if any. So just to wrap up with acknowledgements, I just wanted to say that this proposal that I submitted wouldn't have been what it is only for the time, support and encouragement I received from a number of people, most of them not here. I'm really grateful to Dara for giving me the reassurance that I needed to actually go for a fellowship. And the proposal really kicked off with the willingness of both Dara and Saoirse to meet with me, their enthusiasm about hearing my ideas and allowing me to pick their brains over some tea and coffee. So clearly tea and biscuits are where it's at, but the Earth Institute's already well aware of that. Thank you.